I am professor of Moscow State University of Physics Department. And in my speech, I want to show you the results which were obtained by us even in 70s. In my report, you will find more questions than answers. And I want to show in the large range of concentration of D2O from very low concentrations. These will be the last works of last year, and our former works until with isotope concentration until the pure heavy water, that the interpretation is so complicated, and the isotopic effect itself is greatly dependent on many factors, which will be illustrated in my presentation. So I shall start from the picture, which shows you the hydrolytic activity of sodium-potassium ATPase at enough high temperature, 37 degrees. This ATPase is taken from the bovine brain, and there is some maybe peculiar scale. You see that the ionic strength on this scale is constant because it consists of sodium chloride and potassium chloride. And the points, for example, this is a cross point, belongs to the ordinary water, and you can see this nonlinear curve. And the addition or change ordinary water to heavy water of concentration of 90% and 60% contrast, you can see here, contrast the centers of activity of sodium center and potassium center. And there is absolutely no isotopic effect in the middle of this concentration. We were greatly amazed that decreasing of deuterium concentration to 30% gives quite other curve. And the more pronounced was the concentration of 0.05%. And you can see that in comparison with the control, the influence of high concentration are of similar nature. But the mechanism of action of low dose concentration of deuterium is quite different. Next. This is the linear plot against concentration of D2O. And you can see, usually, all the authors, we have a very similar results with some American results and publications in this region from about 10 to 90%. It may be shown as linear, but it was very fantastic for us. The less we add of deuterium, the more pronounced is the effect. We can't believe to ourselves and didn't publish it for more than 1 year. And check the result many times, but it is appeared and appeared. So the next slide, you see that linear plot is not available in this case. And we come to the logarithmic plot. And you can see that at some concentration, low concentration, we have activation of 150%. And very low inhibition of pure heavy water. The other fact, this is the analogous plot for where the potassium ion is changed for cesium ions. And you can see that the isotopic effect is more pronounced as for low concentration and for large concentration also. That means that hydration effects are very valuable. and the effect of the activation effect of 30% of deuterium disappears, and it lies here. No, this is not a great difference, and we can't differ it statistically. The next one, this is again the ATPase with sodium, potassium ions in the solution. But I want to stress that there is the lower temperature, 13 degrees. Usually with lowering temperature we have the increased isotopic effects. And really we can see that isotopic effects of high concentration are really larger. But the effects of small concentration of deuterium dissipate absolutely. And I want to say that this temperature is correspond to the temperature which is below the phase transition of lipids. This preparation is the preparation of protein-lipid complex. So after this phase transition, when the lipids are rigid, there is no effect of small concentration. We repeat, we were very anxious and began to study. We have, Moscow University have the base, practical base on the White Sea in the north, on the north circle. And there is hydroids. No, this is in small microscopic animals. No, looks like I don't know how to say. No, as hair, for example, on the alga, and we can cut small pieces and study regeneration. And it was amazing that this curve is also has maximum at low concentrations. This is logarithmic scale of concentration of deuterium, and the points with various contrast belongs to different to 2 years in series. You can see that it was very difficult to study very low concentrations psychologically. This is the middle of 1977. So just the same effect as we see in principle with sodium potassium ATPase. At the same time, I want to say that we have the analogous effect on calcium magnesium ATPase, but we have absolutely no effect on the same, from chemical point of view, ATPase of muscle ATPase. There was no effects on low concentration of deuterium. There was no effect at all. Now the other example, it was made several years ago. This is growth of bacteria, and you can see here a very nice picture, maybe the best that we got with other species of bacterium, when you can see that high concentration of deuterium absolutely inhibit the process of growing, and with the depletion of deuterium, we can see that the inhibition is changed to activation at low concentrations. I like this picture greatly. And yesterday, you have heard about the fractionation processes in nature. And this is the example of, at that time, we can't get the really depleted water, highly depleted water as now, and we use the experiment which was provided on drifting ice in North Ocean. And we succeed in this experiment because this is the first experiment in glaciology also. Because when we start this experiment, we fixed the ruler to the ice. And during the ice living, there are several processes. At first, the depleted snow, depleted of deuterium, highly depleted, covers the surface of the ice. But in summer, all this, not all, I don't want to say that all, some part of the snow melts and goes down to this ice. But the temperature of ocean is low, is below zero, but it is salty, so it is liquid. But when the fresh water comes down, it begins to freeze. And so as a result, we can see here a very interesting process as oscillation concentration of oxygen. We measure in this experiment, we measure oxygen 18. And this allows us to, as usual, we get the column from the ice and then cut it on the height and study algae, which is living inside this ice, and with great amount really, and isotopic content by mass spectrometry in Moscow. This is one of the pictures which shows the concentration of blue-green algae measured by the contents of carotenoids just on the ice, and against the isotopic effect of isotopic content of oxygen. And it is very interesting that there are two, I don't know, at first I don't know what to do with these points. And I decided to express it in two linear of regression, except these 2 points. They are very symmetrical, you can see here. It's strange, but I take them off and put this correlation. And I don't know what is the meaning of this point. And on the conference with geologists in Moscow, one of Moscow institutes, they say, why didn't you know? This is the isotopic effect of ancient ocean. So this is also one more question, which the answer I don't know. The same, I want to say that the same was with the green algae also, but the results are a bit complicated. So we come to the hypothesis that the concentration of deuterium oxide and deuterium itself may produce different action. It is very clear that high concentration, this is by no means, the high concentration of deuterium inhibit all the processes, life processes. But in the region of small concentration, there may be activation. But when we began to study water depleted with deuterium, we understand that we were mistaken in general. This is our new experiments with hydrolytic activity. We return to sodium-potassium ATPase, but unfortunately this is not from bovine brain, but from nasal salt glands of a duck. And we can see here that the 24 ppm water produce inhibition, real inhibition, and the 4 ppm water doesn't differ from the original water, from the natural water. And this is the curve for survival of human spermatozoa. And also the result was so that the control that in depleted water, all the process began to be more active, and they are not the spermatozoa have lost their motility faster than in ordinary water. We also make the control with 1.5%, which we produce by adding of heavy water to ordinary water and to light water, the heavy water to light water also, and they are not different. So we can believe our experiment that there is no effect of some maybe admixtures to any portion of water, deuterium or light water. And this is the experiment which shows that development of fertilized roe of loach. No, this is a kind of fish, and we study them during 6 days after incubation, after fertilization. And you can see here that in the water containing 3 ppm of deuterium, the percent of alive cells is twice more than in control. So this result is quite opposite. No, for example, we check this process by adding of 20% of deuterium, and you can see that there is no alive roe during this time. So this is just a very crude control. Now, at the end, I want to express my thanks to the Light Water Company, Russian company, to Tatiana Burdeina and Anton Chernopyatka, which allow me to proceed these experiments with light water. And I think that our experiments will be prolonged, and we shall have more more material and more understanding for this. I want to say that in general, the effects of slow concentrations, and I study water from different sites, and even including homeopathy, it is very difficult to explain, because the large concentration, we have good physical chemical theory for this field. But when you diminish concentration, usually you know this, the discontinuous dilution. Yes. But this is the most interesting field because there is no really, ground base, scientific ground base for many processes in this really region. And you see that activation we can get not only by depleting, but even for increasing deuterium contents in water. But from my point of view, the depleting is better because, and it was not said in the previous lecture, it was said that, for example, in fruits are enriched by deuterium. But our organism is also enriched by deuterium, according to the water which we are drinking. And if we shall drink the depleted water, it will be, I think, better from some general point of view. Thank you for your attention.
Is there a question?
In your experiments, did you correct the levels of other ions or minerals were the same?
Yes, this was made by dry substrate. This is universal thing. There are two variants, absolutely. Questions. I shall answer all of them.
I would come back to the ATPase experiments, which are very interesting data. You've avoided interpreting them. You must have some hypotheses, at least, about
About
how the deuterium is interacting with the enzyme active center. I mean, is this? Do you think this is a physical effect, as to say the size of the deuterium atom, or possibly something to do with the solvation shell for the cation, rather than a classic enzyme kinetic isotope effect? Could you make some comments on that?
Yes,
please?
I understand. No, these effects are complicated and, of course, both. We study also the kinetic isotope effects. I didn't show them to you. And I want to say that it is very productive to study and even the mechanism of enzymatic or any chemical reaction using the mixtures of ordinary and heavy water. And we produce several models. No, I have a few time for a report. And you can see that ATPase reaction in general, they include the proton in flight. But except that, if we have only one proton in flight, we have some symmetrical curve, nonlinear curve, but not so complicated according to the concentration of deuterium. But our curves were more complicated. For example, they have the shape of such kind, rapid increase at the beginning and slower increase at high concentration. And it was explained by the stages of conformational transition. We compare it with the thermodynamic data of denaturation, and the form or the shape of this curve was quite similar from thermodynamics and kinetics. This was very impressive, really. But for low concentrations, this is a great question. I can't answer it properly. So I can make fantasy, but it will be not correct.
And would you agree, and can you confirm, so finally the potassium and sodium ATPase behaves totally different comparing to the magnesium and calcium ATPase, because close to changing the deconcentration to the natural level behaves completely different way, the sodium and calcium, sodium and potassium ATP, but not the magnesium and calcium.
But the mechanism, from the chemical point of view, this is the same reaction, because this is a hydrolysis of ATP. And our preparation was the same also, because we don't study the transport itself, but hydrolytic activity according to the hydrolysis of ATP, by changing of pH.